it's kind of wild to think about how many threats are out there, right?
Yeah.
Like the ones we don't even really hear about.
Yeah.
That can be the ones that are like spreading the most.
Mm-hmm.
And I feel like that's the case with what's going on in Brazil right now. We've got this this fungal infection that's linked to cats.
Mm-hmm.
And it's been kind of silently spreading, I guess you could say.
Yeah.
And it's becoming a pretty significant health problem, to say the least. We're talking Sporotrichosis today.
A silent but spreading threat.
Yeah, exactly. Exactly.
And it is a fascinating deep dive that we have. There is a new study out that's offering some really compelling insights into this growing problem.
Okay.
The research is supported by the São Paulo Research Foundation, FACE-SP, and it's led by Dr. Anderson Messias Rodrigues at the Federal University of São Paulo.
Okay.
And they've uncovered some really surprising aspects of the fungus itself and what might be contributing to its spread.
So Sporotrichosis, for those who haven't heard of this, what exactly are we talking about here?
So Sporotrichosis is a fungal disease.
Okay.
And it typically affects the skin and the lymphatic system.
The lymphatic system.
Yeah, so that's like your body's defense network, you know?
Right.
But in some cases, it can actually go deeper than that. It can affect internal organs as well.
Oh, wow.
And it's caused by fungi in the Sporothrix genus.
Okay.
But what makes this particular epidemic so noteworthy is the fact that it's being significantly driven by transmission from animals to humans.
Oh, interesting.
And domestic cats are a major source of that transmission.
So we're not talking about the kind of like fungal thing you might get at like a public pool or something.
Right.
This involves our feline companions.
Exactly.
So the study really emphasizes the sheer scale of this outbreak.
Yeah.
How widespread are we talking?
Well, this is actually the largest and longest-lasting Sporotrichosis epidemic documented globally. It really took off. It gained momentum in Rio de Janeiro back in the late 1990s.
That's right.
And since then, it's expanded its reach across Brazil and even into some neighboring countries.
Oh, wow.
So the fact that it's been going on for so long and it has spread so widely is quite remarkable and frankly concerning.
Yeah, for sure.
Yeah.
And it's the cats that are really spreading this, right?
Precisely.
It's not like just a coincidental link.
They're really at the heart of it.
Yeah.
So infected cats are really central to how this disease continues to spread.
Okay.
The fungus can be transmitted through scratches, bites, and even through contact with fluids from an infected cat.
I see.
So you can imagine in areas where there are many cats, whether they're stray or pets, the potential for the disease to jump to other animals and to people is significantly amplified.
Yeah, that makes sense. So the study kind of takes an interesting turn and starts looking at the genetics of the fungus itself.
Yes.
It sounds like they stumbled on something pretty unexpected.
They really did.
Right.
They took a close look at 104 different samples or isolates of Sporothrix fungi.
Okay.
And they focused on a specific gene called CMC, which is responsible for producing the GP6070 antigen.
Okay.
So think of the CMC gene as like a blueprint for a specific protein,
Okay.
And that protein is the GP6070 antigen, and this protein is like a unique ID tag on the fungus that our immune system can recognize.
Gotcha.
And what they discovered was a surprisingly high level of genetic diversity.
Okay.
A total of 79 distinct genetic variations.
Wow, 79.
79, yeah.
There's a lot.
That's far more variation than scientists had previously thought existed within this group of fungi.
Yeah, that really paints a picture of a fungus that's not stable. Like what could be driving that kind of change?
Right, and that's where the study gets really thought-provoking.
Okay.
The researchers are proposing a connection between this rapid evolution and exposure to common pollutants found in urban environments.
Interesting.
So they're specifically looking at chemicals like benzene, toluene, and polycyclic aromatic hydrocarbons.
So urban pollution, how could that, car exhaust
exactly,
and industrial byproducts, how could that be influencing a fungal infection in cats and then us.
So the idea is that these pollutants are acting as stressors in the fungus's environment, right?
Okay.
And these stresses could in turn favor the survival and reproduction
I see.
of fungal strains that happen to have genetic adaptations,
Okay.
allowing them to better tolerate these chemicals.
Oh.
Interestingly,
Yeah.
the CMC enzyme, the one they studied the gene for, is also involved in breaking down certain types of aromatic compounds through a metabolic process called the polito-adipate pathway.
Okay.
So it's possible that strains with enhanced CMC activity have a survival advantage.
I see.
In polluted urban settings.
So they're kind of like learning to thrive.
Yeah, it's a fascinating example of how our environment might be directly shaping the evolution of pathogens.
That's pretty, that's a connection you don't immediately think about.
Right.
So beyond just like understanding this, this evolutionary process, do these genetic findings offer any like practical ways to combat the infection?
Absolutely. By pinpointing key B-cell epitopes and conserved glycosylation sites.
Okay.
The studies identified potential, you could say, Achilles heels of the fungus.
Gotcha.
These B-cell epitopes are specific parts of the fungus that our immune system can target to fight it off.
Okay.
Making them promising candidates for vaccine development. And conserved glycosylation sites are like consistent decorations, you could say, on the fungal proteins.
Right.
Because they don't change much across different strains.
Gotcha.
They could also be good targets for new antifungal drugs.
Interesting.
Furthermore, the study found that the CMC gene itself is proving to be a more reliable molecular marker for telling different strains of the fungus apart. Okay. Compared to the markers we've used in the past.
Gotcha.
So this means we could have more precise tools for tracking the disease and understanding how it's spreading.
So a more like refined way to identify and monitor the different hikes out there.
Yeah.
Now the study kind of keeps coming back to Sporothrix brasiliensis.
Yeah.
What makes this particular species such a big concern?
Well, Sporothrix brasiliensis has been identified as the primary driver of this ongoing epidemic. Okay. And it's considered the most virulent species.
Okay. Within the entire Sporothrix genus.
Oh, wow.
So compared to other Sporothrix species, it's more pathogenic, meaning it's more likely to cause disease.
Gotcha.
It also seems to be more efficient at transmitting itself, including the ability to spread readily from cat to cat.
Oh, wow.
Which really fuels the epidemic in animal populations and subsequently increases the risk to humans. Right. Perhaps most worryingly, there's some evidence suggesting it has a greater capacity to develop resistance to the antifungal drugs we currently have.
That's not good.
Yeah, not great.
Yeah.
The researchers also noted the presence of high-frequency derived alleles in S. brasiliensis, which further supports the idea that this species is rapidly adapting and potentially becoming even more virulent.
I see.
Yeah, it's definitely the key player we need to understand better.
So the one to really focus on, it sounds like.
It seems that way. Did the researchers like Dr. Rodrigues, did they offer any of their own kind of perspectives on these findings?
Yes, Dr. Rodrigues stated quite clearly that these findings offer a new molecular perspective on Sporothrix evolution and virulence. Okay. And he emphasized that gaining a deeper understanding of these fungal adaptations at a genetic level is absolutely essential for developing more effective strategies to diagnose, monitor, and ultimately control the spread of Sporotrichosis in both human and animal populations.
Right. So that really highlights why this kind of like deep dive, why this study is so important.
Exactly.
So like looking at the big picture, what's the main takeaway here? What should our listener be thinking about after listening to this?
I think the study really underscores the urgent need for us to step up our fungal surveillance efforts. Okay. Given the continued rise in cases of cat-transmitted Sporotrichosis. Yeah. You know, it's crucial that we improve our ability to track the disease, understand where it's spreading, and identify any new trends or emerging threats. Yeah. The potential to identify specific genetic markers that are linked to increased virulence opens up some exciting possibilities for developing new and more targeted control strategies. Okay. Not only for treating humans, but also in veterinary medicine to try and get a better handle on this growing public health challenge.
Yeah.
You know, it's about understanding the enemy at a fundamental level to fight it more effectively.
Right, like really getting down to the nitty-gritty.
More nitty-gritty, yeah.
So to kind of like bring this all together, we've got, we're facing this pretty significant and persistent fungal epidemic in Brazil.
Yeah.
With cats playing a key role in how it's spreading.
Mm-hmm.
What's really surprising, I think, is this remarkable genetic diversity of the fungus.
Right.
And researchers are exploring a potential link between this rapid evolution and the presence of urban pollution, which might be acting as a selective pressure.
Yeah, exactly.
And these genetic insights, I think, are so valuable.
Yeah. Because
they can pave the way for better diagnostic tools, more effective treatments, smarter control strategies.
Absolutely.
Especially against that Sporothrix brasiliensis.
Right, exactly.
That seems to be particularly concerning. Yeah. And the main message here is definitely a call for increased vigilance.
Yes.
And more robust surveillance of this evolving fungal threat.
Absolutely.
Yeah, I think what's really fascinating for me and hopefully for you listening is this potential interplay.
It is.
Between like environmental factors, like the pollution that we're creating in our cities, and how infectious diseases evolve.
It's a real connection there.
Yeah, it makes you think about how our urban environments might be unintentionally shaping the very pathogens we encounter.
Right.
Yeah, and it's a reminder that these biological systems are deeply interconnected with the environments they inhabit, even the ones that we've built.
Exactly.
It really it really gets the gears turning.
It does.
So like on that note, you know, considering the rapid adaptation we're seeing with this fungus in Brazil.
Yeah.
What other like seemingly localized health issues might actually be influenced by environmental factors in ways we don't really expect, right?
It's a good question.
Yeah. Yeah. It really it shines a light, I think.
It does.
On the interconnectedness of our urban environments.
Yeah.
And the ongoing evolution of disease.
Yeah, a lot to ponder there.
Something to think about.
Definitely.
Yeah. Yeah.
For anyone who wants to kind of delve into the specifics of this research, the study is titled Genetic Diversity and Molecular Evolution of 3-carboxymucinocyclis, PP6070, the major antigen in pathogenic Sporothrix species. Okay. And it's by Jamil Ambrosio de Carvalho.
Okay.
And colleagues. And it was published in Mycology on March 8th, 2025.
Okay.
The DOI, if you want to look it up, is 10.1080215010, who's 03.203.205.2467118.
Wonderful. Well, I think that about wraps it up for us.
I think so.
Thanks for listening, everybody, and we'll catch you next time.
Right. See you then.